When we talk about forensic evidence, our attention usually goes straight to the laboratory. We think about DNA profiling, fingerprint comparison, toxicology, ballistics, question documents, or trace evidence examination. We often picture the forensic scientist sitting in a laboratory, examining an important piece of evidence and finally giving an expert opinion. But there is a part of forensic science that happens long before the evidence reaches the laboratory, and sometimes the success or failure of the entire forensic examination can depend on this stage. A fingerprint can be scientifically examined only if the object carrying it reaches the laboratory without unnecessary disturbance. A bloodstain can be analyzed only if the biological material has been appropriately preserved. A firearm can be examined only if it reaches the laboratory safely and without compromising relevant evidence. A tiny fiber can be compared only if that fiber has not been lost somewhere between the crime scene and the laboratory. So, forensic science does not begin inside the laboratory. It begins at the crime scene and continues throughout the entire journey of the evidence. Once an item is collected, the investigator has a responsibility to protect its identity, condition, and evidentiary value. This involves 5 closely connected processes: packaging, labeling, forwarding, safe transportation, and maintaining evidence integrity. These may sound like routine procedures, but they are extremely important because a mistake at any of these stages can affect the scientific examination and may ultimately raise questions about the reliability of the evidence. Let us now follow the journey of physical evidence from the crime scene to the forensic laboratory. Packaging refers to placing the collected evidence in an appropriate container or package that protects it from loss, contamination, damage, deterioration, and unauthorized access. The most important principle is that there is no single packaging method suitable for every type of evidence. The packaging material must be selected according to the nature, physical condition, and forensic significance of the item. Biological evidence may include blood, saliva, semen, tissue, hair, bones, teeth, biological stains, swabs, and articles carrying biological material. The primary concern with biological evidence is preservation and prevention of contamination. Consider a case involving a sexual assault investigation. Investigators recover a garment that may contain biological stains. The garment is not simply thrown into a common evidence bag along with other objects. The item is handled using appropriate precautions, allowed to dry when required by procedure, and packaged appropriately. Biological evidence that is damp generally requires careful drying before packaging because moisture can encourage microbial activity and potentially damage DNA. Appropriate breathable packaging is commonly used for dried biological evidence. Another important principle is separate packaging. Suppose a crime scene contains a bed sheet, a towel, and a piece of clothing, all potentially carrying biological material. Putting all three together creates an unnecessary opportunity for biological material to transfer from one item to another. Therefore, items that could contaminate one another should generally be packaged separately. This becomes particularly important when DNA analysis may be conducted. A forensic laboratory may be able to identify multiple DNA profiles, but if it becomes much more difficult to interpret evidence when contamination has occurred before the evidence even reaches the laboratory. Therefore, proper packaging protects the biological evidence and protects the reliability of its later interpretation. Blood evidence can appear in many forms. It may represent as a liquid blood sample, blood stains on clothing, blood stains on a weapon, blood on a floor or wall, blood on bedding, blood on other objects. The packaging method depends on the form and condition of the evidence. For example, a blood-stained piece of clothing may be handled differently from a liquid blood sample collected for a specific examination. The investigator must therefore consider the condition of the material rather than simply following a single rule for every blood-related item. This is an important UGC NET concept. Packaging is evidence-specific. Now let us move to trace evidence. Hair and fibers are often extremely small. Their importance, however, can be much greater than their size suggests. Consider a burglary investigation. A suspect is believed to have entered through a broken window. During examination of the window area, investigators recover a small fiber caught on the broken frame. That fiber may potentially provide an association with clothing or another source, but if it is placed loosely into a large bag containing other materials, there is a possibility of loss or transfer. Therefore, small trace evidence needs suitable packaging that prevents the material from escaping or becoming contaminated. Tiny trace materials may be secured in appropriate small containers, packets, or paper folds depending on the nature of the evidence and laboratory procedure. The key principle is small evidence requires packaging that prevents loss and unnecessary transfer. Glass fragments are another important type of trace evidence. Consider a hit and run case. Broken pieces of a vehicle's headlamp are recovered from the road, while investigators later recover broken glass from a suspected vehicle. The forensic laboratory may compare the physical and chemical characteristics of the fragments, but glass is fragile. If fragments from different sources are placed together, they may break further or become mixed. Therefore, glass fragments from different locations or sources should be separately packaged and clearly identified. Rigid containers are generally appropriate to protect the fragments from further breakage. This also illustrates the importance of known and questioned samples. The questioned glass recovered from the crime scene should remain distinguishable from known glass recovered from a suspected source. Soil can also become important trace evidence. Consider a case in which a body is discovered in an isolated location. Investigators recover soil from the victim's footwear. Later, soil samples are collected from several possible locations. The laboratory may compare the questioned soil from the footwear with known samples collected from relevant locations. For this comparison to be meaningful, the samples must remain separate. The questioned sample should not be mixed with the known sample. Each sample should be appropriately packaged and labeled. This gives us another important principle. Questioned and known samples must remain clearly distinguishable. Firearms require special attention because they present both evidentiary and safety concerns. A firearm recovered from a scene may contain fingerprints, DNA, trace material, gunshot residue, ammunition, mechanical evidence, ballistic evidence. Therefore, careless handling can potentially affect several examinations. The firearm should be handled and secured according to established firearm safety procedures. It should be packaged in a manner that prevents unnecessary movements and protects the item and personnel during transportation. The condition of the firearm at the time of recovery should also be appropriately documented. For example, whether a magazine or ammunition was present, the position of relevant components, and other significant observations may be documented according to the circumstances and applicable procedures. The important point is a firearm is both potentially hazardous and potentially rich in forensic evidence. Therefore, safety and evidence preservation must go together. Ammunition should also receive appropriate packaging and identification. Spent cartridges, bullets, and live ammunition may have different forensic significance. A fired cartridge case may be examined for firearm identification, firing pin impression, breech face marks, extractor and ejector marks. A bullet may be examined for rifling characteristics, land and groove impressions, direction of twist, other microscopic markings. Therefore, bullets and cartridge cases should be protected against unnecessary scratching or damage. They should also be separately identified when they originate from different locations or sharp objects such as knives, blades, needles, and broken metal objects present a special challenge. They can injure people handling the package. They can also puncture ordinary packaging. Suppose a knife recovered from a robbery scene is placed inside a thin envelope. During transportation, the knife moves and its point tears through the envelope. Now, the evidence has created a safety hazard, and there is also a possibility of evidence loss or contamination. Therefore, sharp objects should be secured in appropriate rigid or puncture-resistant packaging following applicable safety procedures. The item should be prevented from moving unnecessarily inside the container. Question documents are another category where packaging matters greatly. Documents may contain handwriting, signatures, alterations, erasures, indented writing, ink, paper characteristics, fingerprints, or biological material. Consider a case involving a disputed will. The original document is recovered and requires forensic examination. If the document is folded unnecessarily, written upon, stapled, punched, or exposed to substances that could alter it, its condition may change. Therefore, documents should be protected from unnecessary handling and physical damage. Original documents should generally be preserved rather than subjected to unnecessary alteration. The package should also protect the document from moisture, bending, and other environmental damage. This is particularly important because question document examination may depend on the physical condition of the original document. Chemical and toxicological evidence requires special consideration. Examples include suspected poisons, liquids, chemical substances, drugs, unknown powders, toxic materials. The container must be compatible with the material and should prevent leakage, evaporation, contamination, or chemical reaction. Consider a suspected poisoning investigation. A liquid recovered from a bottle may later be examined for toxic substances. If the container leaks during transportation, the evidence may be lost. If the substance evaporates because an unsuitable container was used, its composition may change. Therefore, the packaging must be selected according to the physical and chemical characteristics of the material. Hazardous materials also require appropriate safety and transport precautions. There is an important exception worth remembering. In fire investigations, investigators may recover material suspected of containing residue of ignitable liquids. These materials are not treated exactly like ordinary biological evidence because volatile substances can evaporate. Appropriate airtight containers may be required for suspected fire debris containing ignitable liquid residues according to applicable procedures. For example, a piece of burned carpet recovered from the suspected point of origin of a fire may contain important chemical residues. If it is placed in an inappropriate package that allows volatile compounds to escape, the laboratory may later detect little or none of the material. So here we see a very important lesson. Packaging must preserve the characteristic that the forensic scientist intends to examine. Modern investigations may also involve electronic devices such as mobile phones, computers, hard drives, memory card, USB drives, and cameras. Digital evidence requires special precautions because inappropriate handling may alter data or damage the device. The device should be documented and handled according to the appropriate digital forensic procedures. For examples, investigators should not casually explore a suspect's phone simply because they have recovered it. The original data may be altered by inappropriate interaction. Therefore, digital evidence should be preserved and examined using appropriate forensic procedures. Once evidence has been appropriately packaged, the next important step is labeling. A package without adequate identification can create confusion even if the evidence itself is perfectly preserved. A proper evidence label may include information such as case number, item number, description, date of collection, place or source of collection, collector identification, submitting agency, seal information, other information required by the specific laboratory. The exact format can vary between jurisdictions and different agencies. The fundamental principle remains the same. The evidence must be uniquely identifiable and traceable. Suppose a laboratory receives 5 envelopes from the same case. One contains soil, another contains hair, one contains glass, one contains a document, and the last one contains a swab. If the labels are unclear, the laboratory may not know which examination belongs to which item. Clear labeling prevents such errors. Labeling identifies the evidence. Sealing protects the package against unauthorized access and helps reveal whether it has been opened. The seal should be applied appropriately so that opening the package would normally disturb the seal or otherwise leave evidence of access. The receiving laboratory can then check the condition of the package. If the seal is intact, then that supports continuity of the package. If the seal is broken or the packaging is damaged, the condition should be recorded and handled according to the applicable procedure. This is particularly important because evidence integrity involves not only the item itself but also the condition of its packaging and the history of its handling. After packaging, labeling, and sealing, the evidence is forwarded to the forensic laboratory. Forwarding is the formal process through which evidence is submitted for forensic examination. The evidence should be accompanied by the required documentation. The forwarding communication should provide information such as case details, description of exhibits, item numbers, seal details, submitting authority, examination that is requested, relevant background information, other required documents. For example, if a suspected poison is submitted, the laboratory needs to know what examination is being requested. If a firearm is submitted, the requested examination may involve firearm and ammunition comparison. Or if a biological sample is submitted, DNA examination may be requested. The laboratory therefore receives not just an object but an object accompanied by its investigative context and examination request. This is very important. The next stage is transportation. Evidence has to travel from the crime scene or police station to the forensic laboratory. During this journey, it must be protected against physical damage, loss, contamination, temperature extremes, moisture, unauthorized access, leakage, breakage, and tampering. The exact transport condition depends on the evidence. Biological evidence may require protection from unsuitable temperature and moisture conditions. Fragile evidence requires protection from vibration and impact. Hazardous materials require specialized handlings. Firearms require secure transportation. Chemical substances may require appropriate containment and regulatory compliance. So, safe transportation means transporting the evidence under conditions that preserve both its physical condition and its forensic value. Consider a burglary investigation. Investigators collect glass fragments from a broken window. The fragments are placed in a container, but the container is not adequately secured. During transportation, the package is shaken. Several fragments break into smaller pieces. Some become mixed with fragments from another source. Now the laboratory received the evidence. The forensic scientist may still be able to conduct an examination, but the original condition and separation of the evidence may have been compromised. This demonstrates why transportation is a part of evidence preservation. Correct packaging can still fail if the package is transported carelessly. Now we reach the central concept of the entire topic, that is evidence integrity. Evidence integrity means maintaining confidence that the evidence has remained authentic, secure, and appropriately controlled from collection through examination. There are several major threats to evidence integrity. Some of them include contamination, cross-contamination, loss, damage, tampering, substitution, improper storage, improper transportation, incomplete documentation. Let's understand them one by one. Contamination occurs when unwanted material is introduced into evidence. For example, a forensic investigator collects a DNA containing sample without following appropriate contamination control procedures. Material from the investigator, another item, or the environment could potentially be introduced. Later, the laboratory detects an unexpected DNA profile. Now the scientist has to consider whether that profile was originally present or introduced during handling. This is why appropriate protective equipment, clean collection materials, and controlled handling procedures are essential. Cross-contamination occurs when material from one evidence item transfers to another. Consider two separate pieces of clothing required from a crime scene. One belongs to the victim, the other belongs to a suspect. If a biological material transfers between them because they were packaged together, the laboratory may detect a mixture that did not represent the original condition of the evidence. Therefore, separate packaging is one of the important strategies for preventing cross-contamination. Evidence integrity can also be compromised simply because evidence is lost. A tiny fiber falls out of an unsealed packet. A hair becomes stuck to the outside of the container. A powder spills because the container was not secure. A document is misplaced among other papers. These may sound like simple mistakes, but they can have major consequences. A forensic laboratory cannot analyze evidence that no longer exists or cannot be confidently identified. Physical damage can also affect forensic examination. A fragile document can tear, glass can break, a cartridge case can become scratched, a digital device can become physically damaged, a biological sample can deteriorate, a liquid can leak or evaporate. Therefore, packaging and transportation must be designed to protect the evidence against the specific risk associated with that item. Maintaining evidence integrity is closely connected with another essential concept which I speak in every video of my physical evidence, that is chain of custody. Chain of custody is nothing but the documented history of the possession, handling, storage, and transfer of evidence. It usually answers several important questions like who collected it, who received it, when was it transferred, where was it stored, who examined it, when was it examined, who returned or transferred it afterward. A properly documented chain of custody allows the movement of evidence to be reconstructed. For example, in this pattern, crime scene investigator, investigating officer, evidence custodian, to transport personnel to forensic laboratory, and then it goes in the hands of forensic examiner, and then at the end it will reach authorized storage or the court. The exact sequence can vary depending on the investigation. The important principle is that the custody and movement of evidence must be properly documented. Consider a homicide investigation where a weapon is recovered from the scene. The investigator seals it and records it. Later, another officer takes possession of it. However, that transfer is not documented. the weapon usually reaches the laboratory. During the trial, questions arise about who had possession of the weapon during the undocumented period. Was it securely stored? Could someone have accessed it? Was the package intact? Was anything added or removed? These questions do not necessarily mean the evidence is false, but they demonstrate why documentation of custody is so important. The chain of custody provides continuity. Let's now put the entire process together. A crime occurs, evidence is identified, the item is photographed and documented. It is collected using appropriate procedures. It is packed according to its nature. It is labeled clearly. The package is sealed. The relevant documentation is completed. The evidence is forwarded to the appropriate forensic laboratory. It is transported safely. The laboratory receives and checks the package. The evidence is stored appropriately. The forensic scientist conducts the required examination. The examination results are documented. The evidence may subsequently be produced or retained according to the applicable legal and institution procedures. At every stage, the question remains: has the evidence remained identifiable, secure, and reliable? That is, evidence integrity. Let's take an example of burglary investigation and follow several different types of evidence. Investigators arrive at a house where a window has been broken. They recover a glass fragment from the suspect's clothing, a fingerprint bearing surface from the window, a fibre caught on the window frame, a shoe impression outside the window, a screwdriver recovered nearby. Notice how different these items are. The glass fragment requires protection against breakage. The fingerprint bearing object requires handling that minimizes disturbance of the latent print. The fiber requires packaging that prevents loss. The footwear impression requires preservation of the impression itself. The screwdriver may contain fingerprints, biological materials, or trace evidence, and therefore must be handled carefully. There is no single evidence-backed solution that can automatically solve every packaging problem. Each item must be considered individually. This is the essence of proper forensic investigation or proper forensic. Let's quickly identify some common mistakes. First, using inappropriate packaging. Second, packaging different evidence items together unnecessarily. Third, failing to properly identify an item. Fourth, inadequate sealing. Fifth, poor documentation. Sixth, improper storage. Seventh, exposure to unsuitable environmental conditions. Eighth, rough or unsafe transportation. Ninth, unnecessary handling. 10th, breaks or gaps in the chain of custody. Every one of these can potentially reduce the evidentiary value of physical evidence. There is one principle that connects the entire topic: package according to the evidence, identified clearly, secured properly, document every transfer, transport it safely, and protect its integrity until examination. This is far more useful than trying to memorize isolated rules, because when you understand the reason behind the procedure, you can answer application-based questions. For example, if UGC NET asks why should certain biological evidence be appropriately dried before packaging, you should immediately connect it with moisture, microbial growth, degradation, possible loss of DNA integrity. If the question asks why should trace evidence be packaged separately, think separate packaging prevents loss and cross-contamination. If the question asks why is a seal important, think security, detection of unauthorized opening, continuity of evidence. If question asks why is chain of custody important, think documented possession and transfer, continuity, confidence in the identity and handling of evidence. So let's follow the complete journey one last time. Collect. The evidence is identified and recovered using appropriate procedure. Packed. It is placed in packaging suitable for its nature and condition. Label. The package is clearly identified. Seal. The package is secured against unauthorized access. document the relevant information and handling history are recorded. Forward, the evidence is officially submitted to the appropriate forensic laboratory. Transport, it is safely moved under appropriate conditions. Receive, the laboratory checks the package, documentation and seal. Store, the evidence is preserved appropriately until examination. Examine, the forensic scientist conducts the required scientific analysis and throughout the entire process maintain integrity. It is very important. Forensic evidence has a journey. It begins at crime scene. It passes through the hands of investigators, evidence custodians, transport personnel and forensic scientists. Eventually, it may reach the courtroom. At every stage, one basic question must be answered. Can we still trust this evidence? That trust does not come from the forensic laboratory alone. It is built through proper collection, appropriate packaging, accurate labeling, secure sealing, complete documentation, safe transportation, and a continuous chain of custody. A tiny fiber can become important evidence. A single cartridge case can connect a firearm to a crime. A fragment of glass can provide an association. A document can reveal an alteration. A biological sample can provide a DNA profile, but all of this is possible only if the evidence reaches the forensic scientist in a condition suitable for the examination. So remember, evidence is not protected only when it is inside the forensic laboratory. Evidence integrity begins at the crime scene itself and continues until the final disposition of the evidence. And that is why packing, labeling, forwarding, safe transportation, and maintaining evidence integrity are not merely procedural formalities. They are fundamental to the credibility of forensic science itself. So if you understood this topic very well, please like, share and subscribe to Criminology and Forensic Science.